recording now. So welcome everyone to our session meeting. We have the pleasure today to have Julian joining us from Taiwan today. She was part of Bi-Chang Chen's lab, but now she started her independent lab in 2020, and she has a lot of experience in tissue clearing, expansion, high-speed imaging, artificial intelligence, analysis, neuroscience, drug delivery, and biochip. So we're looking forward to hear from
you. Okay, thank you. Thanks for Gabriela's introduction. So let me share my screen now. Can everybody hear me now?
Yeah.
Yeah, okay.
I can hear you well.
Okay. So I'll do the screen share. Is the PowerPoint ready now? That's good.
Yeah.
Okay.
Yeah.
Okay, so it's really my honor to be here today. I'm Li-An Chu from National Tsing Hua University. Oh, I just realized I didn't put my school name there. Okay, I'm from Department of Biomedical Engineering and Environmental Sciences in Tsing Hua. And it's my 5th year in the lab. And I started my lab, and I'm really happy to share one of our work, which is the super-resolution imaging in collagen abundant tissue. This is a field that I would say that I never thought I would get into before I start my own lab. So it's really exciting story. Yeah. So a little bit background information of myself. I'm actually graduate from mechanical engineering department in my school back in 2007. So when I just graduated, I think I'm really interested in biology, especially neuroscience. So rather than just trying to get in a professor's lab in mechanical engineering who is interested in biology, why not just join the biology department? So I choose Institute of Biotechnology as my master and PhD degree, and I also spent a year in Cold Spring Harbor lab, and I come back at that around 2018, and I spent 2 years in Bi-Chang's lab in Academia Sinica. So in his lab, I learned all about microscopy, all the lightsheet microscopy, and that's how I do all my experiment today in my own lab. So I really appreciate all the lab members and all the mentor he gave me in the past few years. Okay, so this is my lab team. We do a lot of bioimaging, and it's kind of natural to have these 3 groups in my lab, like develop independently, but also collaborate with each other. So we have this tissue engineering team who's the leading students are like working on tissue clearing, multiplex labeling, and also the expansion. So most of the work will be done by that I will introduce today is done by Yahan. And also we have these microscopy team who so the student here is focusing on developing new microscopy. For example, we recently built a near IR light sheet high resolution light sheet microscopy that we're gonna use to image some near IR molecules that are able to show the vesicular system, but also allow us to do the treatment at the same time. So it's very exciting. And also they built the microscopy that allow us to do all the large sample imaging, including the expanded sample. Yeah. And after you acquire all the like multiplex image, the most important part is data analysis. So because all the user here once upon a time already suffer from like large data size, right? So we get this terabyte data every day. So it's almost impossible to do the analysis manually. Well, some you can, but mostly, for example, the whole brain data, you just can't do it with, manual segmentation. So deep learning team is kind of a natural option to analyze all these data. But so we're not focusing on that part today, but if you're interested, I'm more than happy to share about how we do the entire process, maybe in other occasion, or we can talk in, schedule a meeting separately. Okay, so the motivation of do this whole brain, the tissue clearing on whole organ is because we wanted to see what happens in the brain in a more complete way in a global perspective, especially in some in the neuroscience research, or for example in my school, in my department, a lot of people are interested in how to do precise drug delivery. So both fields highly needs the the more complete view of the like the brain system or the intestine system, or like lung or stomach, all different kind of organ could be used in their independent studies. So in my lab, we are trying to so most of them are using like MRI to, or like CT or IVs to visualize the entire body, but also use a lot of electron microscopy to visualize detailed structures. But of course, the microscopy can, most of them are using microscopy in their lab too, but they may only like small chunk of the sample. So that is really not giving us too subjective view of the data. We know how easy to choose the right part to analyze, right? So so that's why my lab is focusing on these large tissue imaging, and also like trying to use the same equipment, same data processing, same sample processing way, but allowing to also push the resolution reach the EM level. Okay. So apparently the traditional confocal microscopy will not give you enough imaging speed to image the expanded tissue or the cleared whole brain tissue. So in my lab, we mostly use the spinning disk microscopy, which is multi-point confocal, or the I think majority of data was taken on with the lightsheet microscopy. So you can buy all this microscopy, of course, but we think it's actually easier that you can build your own instrument, because sometimes you just need a specialized form of sample holder or specialized light pattern to get a better image. So this is how we kind of assemble our own microscopy in the lab. And it's pretty fun, and you can actually recruit more students if they, from more engineer students to come to the lab, because they like to play with these toys. Okay, so in the past 2-3 years, we have a very successful collaboration with a lot of different amazing scientists in biomaterial field and neuroscience field. So most of them are focusing on the large sample image like this. Sorry, I have to move the okay. So but just last year we started the collagen abundant tissue, so the expansion protocol specific for the collagen abundant tissue. And here's why. Okay. It's kind of not working. Okay. So before I start my own lab, the work I did in Bi-Chang's lab is using lightsheet to do localization microscopy, not expansion at all. Because by then it was like 2016 and expansion just, just published. The first expansion paper was published in 2015, and not really a lot of people aware that's gonna kind of, I would say, taking over the entire super resolution field. But back then, we believe that localization is also really a powerful tool that allows to see neural structures and detailed neural structure in animal brain. So we did have some great data that showing us the localization process can help us to reconstruct the neural structure like what you see on the screen. And it gave us a pretty nice, like better resolution compared to the traditional light sheet. But the problem is, there are several problems actually. So the most important one is it's really hard to get multicolor. And also like the sample preparation is a little bit tricky. You need a specific dye for the labeling. So that's another reason why, that's actually the main reason why it's really hard to do multicolor. So in 2020, then we started say, okay, maybe we should shift our focus to expansion. And we had some really good data on Drosophila brain. And this is the expansion data from Drosophila brain. You can see the neurons and the synaptic vesicle directly. You can even count them, like count how many synaptic vesicle on these neurons. So this is really promising, and we tried to apply that to human brain, and it's also very successful. This is a 1 millimeter human brain block, and we're able to see all the the exercise and neurons and the vesicular system all together and see how they like interact with each other. These are all done by traditional expansion protocol. We did, we don't have to modify a lot. Mostly just, how many days for maybe longer time for the monomer penetration, that's all. But so we published this data about collagen abundant tissue because the traditional expansion protocol doesn't really work on this type of tissue, the collagen abundant one. What is the collagen abundant tissue? Here I listed some of them, that like the skin fiber, and also like the pancreatic tumor that which is also highly collagenized, and then also like hair follicle. These is also belong to the skin parts, and also the corneas. So all the peripheral system, like the skin, like blocking the water get into your body, they have a lot of collagen there because they also wanted to maintain the strength but softness. Like it can be stretched, but they still have maintain some of strength to keep all the hazard material get into your body. So before I get into the detail, I just want to show you two images that are showing the power of expansion microscopy on this particular tissue. So this is a collaboration between me and our lab and Professor Ling Songran's lab in National Taiwan University. He's a dermatologist. Yeah. So you can see that with expansion, this is after expansion image, you can see very clear neuronal system around the sweat gland. So this was really hard to do in the past with even 100x objective under the regular microscope, because you can see how how these cells aggregate together and then become a very dense structure in the sweat gland. And also the hair follicles, like the whisker hair follicle, you can see how dense the structure is. All these data are taken after expansion. Otherwise, you'll see all the cell will, kind of mix together, and you won't be able to even just count how many cells are there. Yeah, and there are a lot of skin-related and hair-related disease that rely on this technology now to really reveal the detailed changes in the neural system around the these structures. Professor Lin Songren actually published a Cell paper a few years back that they found the dopamine system is the key to allow you to have the goosebump. Very, very cool. And we are collaborating on more project now to see if we can find more new structures and new neuronal systems in the skin. So here's the a very small literature review that so most of the expansion protocols that are being applied to tissues and cells are only apply to very relatively thin sample like under 100 microns, and so and most of these tissue that being applied to have relatively low collagen percentage, like collagen content. So like the brain is lower than 3%, and kidney is around like 5 to 10%. And here maybe the pancreas maybe a little bit higher, and the colon maybe a little bit higher. These are like 15 to 20. And even just get to these sample, you really need a very harsh digestion, and then you can only apply. Well, maybe they haven't tried Seeker 1, but the sample they demonstrated is only 5 microns. But in order to do image like this, you really need a protocol that be able to apply on millimeter or like 500, at least 500 micron thick sample, and the collagen content of skin and cornea is above 70. So there's a huge difference from 15 to 70. Okay. So that's the goal. And long story short, we played with the digestion and the digestion process, and both the temperature and the collagenous. But I have to be honest that we later figure out not all the tissues really require the collagenous. Not all the collagen abundant tissue really require collagenous to do the Foley digestion. But I think the key player we did here is still the protein escape. But so my student have a series of test of how long you should put their sample in protein escape, what's the temperature you should use. So like play with these parameter a lot. So you'll see that 24 hours may be good enough, but usually we will recommend like 48 hours to fully digest all the proteins in this collagen abundant tissue. And so here is 2 examples. One is the cornea and the other is dorsal skin tissue. And you can see the enlarged data below here. So you'll see the cell shape maintains really well, and even the hair follicle structure maintains really well even after 48 hours digestion. And we don't see a lot of expansion error in this tissue, so we think it's good to it's okay that we can use this data to do some kind of further analysis on our sample. And so here are several more example, like so in the cornea we can see there are different layers of tissue, different layers of cells, and in the paper we mentioned that the expansion microscopy can enhance the transparency. I think most of the people in the expansion group might think it's kind of a straightforward result that you always get clear. Of course, you get more transparency after expansion. But the reason we kind of point out this issue is that so most of the brain tissue, or let's say lung tissue, before you do the expansion, you can also use the tissue clearing to make it fully transparent. And if you don't need such high resolution, you can easily image the entire brain. But with the collagen abundant tissue like the cornea, skin, and hair follicle, the tissue clearing never really worked well. So we never really get a very clear image. Of course, we have to do the immunolabeling compatible way. So we didn't try the organic solvent way to clear the skin, but if it's a aqueous-based tissue clearing, then you can never get these tissue clear enough to see all the details through the entire structure. So that's why we think using the expansion can also give us a lot of benefits to see more clear structures through the entire like different layer of these tissues. Okay, so here are just some measurement of how transparent and how not transparent of this. I think this is a skin sample. Yeah. Okay, so another very interesting question is that can we maintain the collagen structures after the expansion? The answer is no, yes or no. So we found that actually so most of people image the collagen fiber with SHG, the second harmonic generation image. So after expansion, all the SHG signal is gone. So if you're really interested in how collagen fibers formed or like how they organized, then you should label them. And we found our protocol is really compatible with all the kind of immunolabeling. So once you can label all the collagen fibers with antibody, then you can still see them very clear after the expansion. And I think this is really useful for the people who study like cardio, like fibrosis problems and like how like the wound generation, like wound healing problems, because they always really care about how the collagen fiber organized and after regeneration. Yeah. So because I was trained as a neuroscientist in my PhD, so I was, mostly interested in neurons. So we're trying to see all the neurons in different collagen abundant tissue. And we do see something very interesting, like there's no reference, literally no reference showing that there are synapses on the animal, inside the animal cornea. And we found some synapse-like structures. We don't know if it's synapse yet because we haven't labeled the synaptic marker, but we do see some of these tiny structures that you can never see with the traditional microscopy. And so the neuron system in the eye, in the cornea, is really different from the one you see in the brain. And you can also see these like little holes in the neurons, and we believe they are mitochondria, and we use another transgenic animal to prove it. Yes, there are mitochondria's home, and it's kind of very interesting. You can see that the diameter is actually different. Like it tells us that the mitochondria size in different organs seems to be different. So we don't know what's that mean, but it's a very interesting topic for the people who study the metabolism, because the mitochondria is the source of energy, right? So it's really interesting that naturally they grow in different size and different arrangement, and why they're arranged like this is maybe highly correlate with their function. So we don't know that, but apparently with expansion, you can allow people to see this structure directly, even without labeling the mitochondria. Okay, so the last application we did is to visualize the tight junction. So all the skin or skin tissue or cornea or all these collagen abundant tissue usually have a lot of tight junction structure there because they're trying to, separate different environments. So we use a cornea as our sample, and then we tried to see if we can visualize the tight junction structure and the affecting the cytoskeleton inside this, along with the tight junction. And compared to stat image, we tried the stat imaging first, and you'll see that most of the tight junction signal are highly correlated with the cytoskeleton, and which is a little bit weird because according to other reference, according to other reference that actually there's only 28 amino acids in the cytoskeleton structure that attach to tight junction. So these data are really confusing, and that's why we think maybe expansion can help. And after we expand the cornea in the middle part here, you can see actually the tight junction and so the cytoskeleton is actually, become like track shape, and the tight junction is aligned in the middle. So it's kind of like the cytoskeleton is pulling the tight junction from both cells. On the left, this is a cell, and on the right side, this is another cell. So to prove that these data can show us how the tight junction actually work and what's the balance to keep the tight junction there, we apply EGTA to disrupt the tight junction, and we want to see how the cytoskeleton actually change. Okay, so this is the data that we apply EGTA on this cornea tissue for 2 hours. So during this process is very interesting. So in the beginning, you can see the ozone is very intact, and then having this very beautiful shape, and all the cytoskeleton is aligned with the tight junction signal, but also there's a radial shape affecting signal that kind of project from the center of the cell. So the idea is that these orthogonal cytoskeleton can kind of pull the tight junction from each side of the cell, so they can maintain the structure for each cell. But after you treat the EGTA, the tight junction kind of starting to fall apart. So you can see there's gap between cells, and then these tight junction are like from 2 different cells are separating with each other. And now you can see the radial structure of the cytoskeleton is still maintained after 30 minutes, but they are all gone after 1 hour. So so now you can see the cytoskeleton structure start to kind of randomized align. They don't align together anymore, and they kind of like more co-localized with the one. So you can see the image here. They are more co-localized at this stage. And in the end, all the ZO1 structure are kind of, disrupt and, align, like separate, like kind of separated in the entire cytosol. But the affecting, which is very interesting, go back to the cell membrane. They don't attach to ZO1 signal anymore. So you can see in the merge data here, they don't align with the tight junction anymore. They kind of go back to the cell membrane. So it's a very interesting process. We are thinking that maybe this could be used to help some of the doctors to see if their drug, how like if can use this kind of mechanism to get more penetration to the into eye or into skin. Okay, so this is just a schematic figure that about what we saw with expansion data. Okay, so this is the last demonstration. So we did try this protocol on the pancreatic tumor, and we do see great result that after expansion, you can see a lot of vesicular system like around these cells, and which can never be seen with pre-expansion data, because the pre-expanded tumor, the cancer tissue is really dense, and you can't really distinguish all the details in this type of tissue. Okay, so that'll be all for the expansion part. And just want to take like 5 more minutes to talk a little bit about other things we do in the lab. So for the tissue clearing part, it's been developed for over 10-15 years now. So there are many ways to do the tissue clearing, but we figured that there are lacking of protocols that allow you to do on formalin fixed paraffin abated the FFPE tissue. And we just, we have a lot of doctor friends working in the hospital, so they will keep continuously asking us, can we do that? So we try on the mouse brain and it works. So we developed this heat clear protocol, allowing you to retrieve the entire mouse brain and do the tissue clearing from the paraffin embedded mouse brain. And this is the simple, the schematic figure of the protocol. But basically, this protocol is involving a specific way of antigen retrieval, so that we can label all the antigen and do the tissue clearing afterwards. So this is just a very short example that's how we apply this tool. So recently we have worked with some my colleague who works on biomaterials and to try to treat wound healing. And we did try several runs of immunolabeling on the same sample and works really well. So the difference between the freshly fixed sample and the formalin fixed sample is that it allows you to do multi-run of labeling through the entire brain easily without any kind of tissue expansion, and then you kind of keep them in the same shape. And to prove that, and the other advantage, so this is the first advantage. The second advantage is that you can actually do this protocol on formalin fixed tissue, like even after 3 months of formalin fixation, you can still retrieve all the antigens. And the correlation between this project and the expansion is that we figure if you have a like FFPE sample is in millimeter size and you wanted to do expansion, this is a pretty good way to do the pre-processing. So after heat clear, you can label the tissue and then you can do the expansion directly on the millimeter sized sample. Not like, so we tried EX path and it didn't work on millimeter sized FFP tissue, but after heat clear pre-treatment, you can do it directly. Okay. So this is the another interesting direction that we also developed some deep learning way to make the microscopy image more isotropic. So we call it digital super resolution. So you can see here, it's more like a deep learning way to do deconvolution. So we all know the axial resolution is much poorer than the lateral resolution in microscopy. But my genius colleague Gary he's able to make this generative model that So now making the entire image become more isotropic. So you can see these two are the same image, but the right one is being enhanced with deep learning, and all the small structure of the neural system is maintained. You always we always get the question that how do you know this is real? So we're working on it. We are using our lightsheet to do this multi-angle image and to double check that how close the image will look like with deep learning and with the real camera, real lightsheet image. Okay, so I want to thank for everyone's attention today, and the talk, the content today is mostly about the technology development, that how we play with the sample and then making them more useful in different fields. And in the future, I wish we have more time to talk about the neuroscience study and or the drug delivery study. They are really cool that we can really see how this imaging technology benefit these 2 or 3 different fields. And okay, so this is the talk, and I want to thank all the students, especially Yahan, who is mainly the key person. She's the key person to the entire expansion project, and Yahui is focusing on the large sample tissue clearing, and Zhang Hen is the leading student who works on the microscopy part, and Li Wen is focusing on how to analyze the data. And Yingxu, they are both computer genius, so they can, we just have to tell them which kind of data we want to show, and they will try to do all the segmentation and or the automatic analysis. So they are really great students. Okay, I think that'll be all. Thank you.
Thank you, Julian. That was great. If there are any questions, you can put them in the chat or you can unmute yourself while people think about their questions. I had a question about more like the light sheet you guys are using. Is that a home built light sheet? Can you give us a bit of details about what's like?
Okay, so so we actually have 2 models in the lab. So one is this one is more the axial swipe light sheet. So axial swipe light sheet is easier to use, easier to build. They don't have the gavel system that allow that you if you want to swipe the light beam into a light sheet. This is more like a steady light sheet that you only move your sample. So I do think if you're so I don't know if that's the information you need.
Yeah, like how large can the sample get, for example?
Okay, so so that's the beauty of these kind of setup. So using axial swipe light sheet, usually we well, I would say that most of the for example the mega spin, I think a lot of people heard mega spin. They also use the axial swipe approach to image like entire mouse, or it can be up to like 2-3 centimeter. I would say like maybe 3 centimeter is the maximum of this method, but that's for the clear tissue. I mean like non-expanded tissue, because no matter how much clearing you did, how clear you think the sample is, it will never become, the refractive index will never be exactly the same as the environment. So I would say like 2 centimeter will be the maximum size for each axis. Oh, of course, in the axial axis, it's really depending on the objective you use. But if you use the expanded sample, then usually the size will be different. Will be no constraint. So the biggest expanded sample we image with our system is around 6 by 4 centimeter. yeah, it's quite big. Yeah. And the hardest part is how to maintain the fluorescence. So I think the environmental control is quite important. Like you don't want the you don't want any kind of disturbance, and you don't want light get into the system, and you want to make sure that the laser power is just enough for the imaging, but not too strong for photo bleaching. Yeah. So and it's also very important to choose a good detection objective, because we know how important the how valuable the photo budget is in the expanded sample. So try to get a higher NA objective is also always a good option. Yeah. Yeah. I mean, the basic principle is always the same, but the reason we choose the axial swipe to do large sample imaging is usually because it's easier to handle the large sample. But if you're doing like cell expansion, like after expansion, maybe your tissue is only your sample is only like 200-300 microns, then the scanning microscopy will be better, I think, because using, for example, this is the scanning microscopy schematic figure. And the reason why I said this is better is when you move the expanded sample, if you don't put the argos or make like try to make it harder, usually the vibration will kills the data. So but with these kind of scanning microscopy, it's easier to do the laser scanning, so your sample don't have to move, so you just move the light and move the objective you're using. Yeah, so it's really depending on like what's the purpose. If you want to get a larger field of view and larger imaging site area, or you want to focus on small area and you really need the high resolution data. Because if you do like large sample image, sometimes a very small vibration doesn't really hurt the data quality. Yeah. Yeah.
Thank you.
No problem.
We have a question. You want me to read it out? So it says, great talk, thank you. Regarding the big image data analysis, what commercial or open source software do you prefer? Do you have any suggestions on how to handle the big data?
Okay, yeah. So I don't know if you refer to the images stitching or the analysis. Can you? Yeah, they are like two different issues.
I think it's a big image data analysis, is that saying?
Data analysis. Okay. So so the major task we do in the lab is imaging segmentation, right? So you want to binarize the data and then quantify that how many pixels are positive signal, how many pixels are negative signal, or like we wanted to calculate the vesicular system, like what's the branch number, what's the yeah. So I would say that UNet in deep learning is usually good enough, and there are a lot of open source example for UNet install, and it's a Python based code. So I think most of the computer is good enough. But if you don't want to get into that so quickly, you want a good user interface based. You want to use the tool that already have a user interface. The ImageJ based tool like CellPulse and like DeepLabCut. The LabKit and Stardust. They are really good tool and really useful. We use them all day, every day, to process a relatively small region, because those data is really good when you only focus on a part of your sample. When I say we have to use UNet or NUnit or any other kind of deep learning, it's really because we're trying to do like whole brain or like whole column or like whole lung. So there are a lot of fibrotic system or fibrotic signal that have the same intensity as your data, but you don't want it. The good thing about expansion is that usually those, unwanted background signal are gone. So that is really a big advantage of doing expansion microscopy. So all the analysis tasks should be easier. And I would say that with higher expansion factor, usually it's more easy, like 10x or 20x expansion. You get very good signal background ratio. So you don't have to worry about like how to train the model. Usually the lab kit and there are weak counts, and then what else? Yeah, they are all really good. And then after you acquire those binarized data, and well, I have to say that then you have to look for the reference and see if uh, there are similar, like there are existing code for you to deploy. But I'm not a computer scientist, and I would say that these tools are really user friendly. So don't be afraid to look out for the image, especially ImageJ based forum. And there's like Deep ImageJ Zoo that you can check on, and all the 3D image rendering that all the movie we make are made with Immerse. So Immerse Viewer is free, and you can do it. And recently, we will switch the entire 3D viewing part to Napari. And if you are, you're gonna work on large data size from now for a very long time, I would strongly suggest that Napari is a forum that I would say it's a community that you want to join, because it's a platform. Napari is a platform that it already have a lot of pre-installed software that allows you to play with big data, and you can kind of set up a server on your own lab, so other people can access the data directly with the Napari format. And the data format they are using is ZAR, ZARR, and is much better than the TIFF because it kind of chop your data into small pieces. It's a pyramid data format, so you can zoom in and zoom out the data very quick. Yeah. Sorry, there are a lot of jargons there, I know. Yeah.
Thank you.
No
Any
problem.
more questions? Mm, Well, I was wondering about the HIF clear process. Is that,
Yeah.
I'm assuming it's a water-based.
Yes. So it is a water-based one, and we do regular immunolabeling after the clearing process. So so basically it's compatible with all different kind of antibodies.
And then the imaging solution is like
Also
a,
the regular one, like you can use anything you want. Like the one we use is developed by a Taiwanese company called wait uh NFC. Yeah, that's the one we're using now, but you can also use like easy clear or like anything that allows you to do R matching, it works. Yeah, the main developing part is to optimize the AR part, the antigen retrieval part, because it's easy to do on thin slice, but it's relatively hard to do on whole brain level, like centimeter sized tissue. Yeah. And I think the good part of using this is you can always embed your sample for a very long time before you know what kind of antibody you want to label them. So we're trying to, sell this thing to the brain bank, because in Taiwan, we have a new brain bank there, and they always put the sample in formalin like forever. But that's bad for imaging afterwards. But the purpose to put in the formalin forever is the same as put in the wax. They want to preserve the sample, making sure they don't get um get hurt or like deformed. But yeah, it's a little bit hard to try to waxing the entire human brain. So we're trying that part for now. Yeah.
That's very cool. I don't see any more questions on the chat, and if anyone wants to ask anything or unmute. Another question from Wei-Chen. Do you save the image into SAR format
Oh, I saw it.
when imaging acquisition, or do you need to perform stitching and TIFF and then convert into SAR?
Oh, yeah. So that is a good question. So for now, we save in TIFF and then do the stitching with TIFF. But I think within 3 months, we will switch to SAR directly, because it's really depending on the camera system and how you write the code into the to the computer. I will say that I don't have the best answer, because I'm not I don't write the code by myself. So I can't really give you the absolute answer when the data become ZAR format. But what I know is after stitching, they will be saved in the ZAR form and then do the processing. So before stitching, I really don't know. I'm sorry. Yeah, I should ask my students. Yeah, thank you.
Okay, thank you. I think no one else has any questions. That was great.
Thank you.
And if people want to get in touch, they can send an email to Julian with more questions, maybe about the data part.
Yeah. I mean, data processing is always, really pain in this entire field. It's like we generate too much data every day.
Yeah.
Yeah.
Thank you very much for that.
Thank you.
Okay.